Okay, it looks like we've got a good audience here. So I'll kick off. Welcome everybody to the monthly Genomics England research seminars. We've got two excellent speakers today who I'll come to, but next slide, please. First thing back up. I just wanted to bring to everybody's attention was, for those of you who are following the Genomics England blog post, there's a new blog about researcher on the rise, Alfredo Duenas-Rey, I hope I've pronounced that correctly, who's going to talk about his experience using the National Genomics Research Library to map genetic causes of rare inherited eye diseases. The Research Network newsletter will have updates about this and other blogs, no doubt. And if you're not receiving that already, then please reach out to us and we'll put you on the mailing list for it. Next slide, please. So we have, as I said, two excellent speakers this afternoon. There's who I will introduce fully as with their talks. But first up, Jayne Barbour, who's going to be talking about cancer analysis of CTCF cohesion binding sites, and Máté Manczinger, who's going to talk about something that's a bit closer to home for me, which is about synergy of HLA class 1 and 2 alleles and the strength of anti-tumor immune responses. Next slide, please, before we start the actual presentations. Just to mention that we won't be having a research seminar in December, but we will be restarting again at the end of January. So the 30th of January, 2 o'clock, last Tuesday of the month, we'll be having another seminar and we'll send the speakers' details out when we have those. So over to the speakers. The first speaker is Jayne, who's going to present about a pan-cancer analysis of mutations in CTCF binding sites reveals a role for uracil incorporation and repair by XPD, which of course is, everybody knows, is the xeroderma pigmentosum D component, correct?
Yes.
Right.
Yes. So I'll talk about, some recent work on XPD, plus I'll also give the backstory of the long story of our pan-cancer analysis on CTCF cohesion binding sites that got us here. So our group is particularly interested in patterns of somatic mutation in cancer. So one way I mean that is by the signature, which is the 6 different SMV types classed by the nucleotide on the 5' and 3' end. Giving a distinctive pattern, such as signature 1 is associated with CPG mutations. And the other way we're interested in patterns is the uneven distribution of mutations genome-wide. So, for example, it's largely governed by epigenetics. Late replicating heterochromatic regions tend to have more mutations, whereas active chromatin and active genes have less mutations. And you can also get these regions of hotspots, which will be very at a fine scale localized increase in mutation density in transcription factor binding sites and CTCF cohesion binding sites, which I'll talk about today. And what's interesting is the pattern of the signature a sample has is usually associated with the pattern of the heterogeneity of mutations across the genome it has. And for example, my supervisor previously published that signature 7 cancers. So this is an ultraviolet light signature displayed in skin cancer. They have mutation hotspots at CTCF cohesion binding sites. So a little bit about CTCF. It's a transcriptional repressor protein that binds to a very specific motif, but it also has a role as an insulator and in DNA architecture, because when 2 CTCFs that are distal kind of dimerize in a complex with the cohesin complex, you get DNA loop anchors. and it's specifically at these DNA loop anchors we see the mutation hotspots. So a long time ago, we wanted to identify cancer samples that have elevated somatic mutation densities in CBS and relate it to mutational signatures and processes. And at the time we used the PCOG cohort, we found 1,980 whole genome sequence samples that had enough mutations to do this kind of analysis across 20 cancer types. And we analyzed the mutations falling in CBS and clustered by mutational signatures. So this is the analysis of the CBS enrichment. You can see esophageal adenocarcinoma and skin cancers have the highest median CBS enrichment. And there are some very high liver cancer and bladder cancer samples, which I will talk about. The orange samples we decided are enriched by very stringent criteria. And then on these enriched samples, we did de novo mutational signature analysis and clustering. So one cluster is just an unusual melanoma sample we don't have an explanation for at the moment, but one cluster looked a lot like signature 7, and they were all skin cancer samples, which is great because it's consistent with our previous work. One cluster was associated with signature 17 in gastrointestinal cancers, and that's mostly esophageal adenocarcinoma. And there was this other cluster of signature 5 mutations, and a few of these samples had XPD mutations. But the signature 7 and 17 have previously been reported, so we wanted to focus on this cluster of signature 5 and XPD mutations for now. But we also looked at the pattern of mutations across the CTCF motif, and we found that signature 7 cancers have a distinct pattern of mutations across the motif, and the signature 5, signature 17 cluster, they share a similar pattern, or exactly the same pattern, which suggests a common mechanism between signature 17 and XPD mutant samples. But at this stage, we had no idea what it was, but I think we figured it out. I'll show you as we go. So at this point, we wanted to focus a lot more on XPD, and XPD is in cancer, in the context of cancer, is mostly mutated in bladder cancer. So I'll talk about the mutational processes in bladder cancer. Other than signature 1, we have signatures 2 and 13, and they're both associated with ApoBec activity. ApoBec is an enzyme that deaminates cytosine to uracil in TCN trinucleotide contexts. So uracil can get replicated as a T, causing a C to T mutation, or it can get excised by uracil glycosylases, leaving an A basic site, which can be filled in by different polymerases, causing different errors and hence 2 different signatures. And then signature 5, which I already talked about, has previously been published to be associated with ERCC2 mutations, and that's simply the gene name encoding XPD. So a little bit more about it. So about 10% of bladder cancer samples have missense variants in XPD. They're annotated in OncoKB as oncogenic. It's interesting, they're never truncating mutations. The protein is a DNA helicase protein, and we typically see point mutations in helicase domains. and the protein is involved in nucleotide excision repair. And DNA repair deficient genomes generally display a very interesting and unique genomic distribution of mutations, which we like to study to learn more about how the DNA repair is working. So the aim of this study was to explore the consequences of XPD mutations on genome-wide somatic mutation distribution. And here we used the Genomics England cohort of bladder cancer samples. So PCOG only has 4 XPD mutant bladder cancer samples and 3 mutant liver cancer samples. But by using Genomics England, we got our numbers up to 39 XPD mutant bladder cancer samples and 343 wild type. We had a further 10 samples which we did not classify as either mutant or wild type because they had protein damaging mutations in XPD, but they weren't recurrent. So we weren't completely sure, but I apply a model to them later, which I'll show you. And then we wanted to dissect out APOBEC from other mutational processes. So this is quite straightforward because APOBEC is so specific to TCNs. I just separated mutations as TCNs or not TCNs and called them APOBEC or other. And then for some analyses, we can further break the APOBEC into SBS2 or SBS13 based on whether they're C to T or C to R mutations. And the methods are quite simple. We just counted the actual somatic mutations falling in a given region, which we call observed, and the expected, which is the background mutation rate from 100 trinucleotide preserved shuffle simulations. And we get an observed expected ratio, which is essentially the mutation density of a particular sample in a particular region. To begin with, I looked quite globally and I calculated mutation densities in 1 megabase windows genome-wide and did principal component analysis. And you can see separation between the XPD mutant and wild type samples, indicating a difference. So I looked into more detail of those factors that affect mutation rate I talked about earlier, such as replication time and chromatin accessibility. And you can see that XPD mutant samples have a redistribution of mutations away from late replicating regions towards early replicating regions, and similarly away from closed chromatin towards open chromatin. This was particularly noticeable for APOBEC mutations with regards to replication time. So we looked into more detail at the APOBEC mutations, and we found, interestingly, XPD mutant samples had more signature 2 and less signature 3. So that means more C to T mutations at TCNs. And it was these signature 2 mutations that displayed this really strong replication time phenotype. And we wanted to look into more detail at DNA's hypersensitive regions, and in particular, of course, CTCF cohesion binding sites, or CBS. And we're really happy to see that the Genomics England cohort it showed exactly what we were expecting based off our small study in PCOG. So XPD mutant samples certainly have strong elevation of mutations in CBS. So we hypothesize XPD is repairing some sort of error or damage at CBS. So I accessed XPD ChIP-seq data And it is bound there, suggesting it could be fixing something there. Finally, we wanted to see, actually, can we use all of this genomic mutation density information of all these different epigenetic features to predict whether a sample is XPD mutant or not. So we trained a support vector machine model, and using leave one out cross-validation, we had very high accuracy. Actually, our model predicted only one wild type sample as mutant. And when we looked at that sample manually, it had a very, very low allele frequency XPD mutation filtered by the variant caller. And we got 35 out of 39 mutant samples. It was completely accurate for PCOG. And the most important feature in the model was other mutations in CBS, which is not surprising as those mutation hotspots are so strong. And then I applied this model to the 10 unclassified samples I talked about earlier. And the model predicted 8 out of the 10 to be XPD mutant. And we looked at the location of the mutations in those 8 samples were all in conserved regions of the helicase domain. So we wanted to understand what XPD is actually repairing and what the mechanisms of CBS hotspots were. So to make a long story short, we noticed XPD mutant bladder cancer mutation patterns mirrors that of uracil glycosylase deficient cells. So we hypothesized it has a role in the repair of genomic uracil. And I mentioned that cytosine can get deaminated to uracil by ApoBec, the process that happens in bladder cancer. But interestingly, uracil can also get incorporated into the genome by DNA polymerases in place of thymine, forming AU pairs. And uracil from AU and GU pairs are excised by the same uracil glycosylase. But this is really interesting to us because 5-fluorouracil is a thymidylate synthase inhibitor, hence increasing DUTP levels in the cell. That's been shown to cause signature 17. And I said before, signature 17 is associated with the CBS hotspots. So I hypothesized uracil is getting incorporated into CBS, and that's underlying the mutations. So I accessed base pair resolution uracil incorporation seq data. And here are the results. The top panels are the somatic mutations in bladder cancer, and the bottom panels are the uracil incorporation. You can see uracil does get incorporated into CBS, but what was really exciting for us was if you look in detail at the motif, it's in the perfect positions to be in line with mutations. So to summarize, XPD mutant bladder cancer have a distinct distribution of somatic mutations. And we hypothesize it's involved in the repair of genomic uracil. Uracil incorporation into CBS likely underlies mutation hotspots there. We think in XPD mutant cancer and in signature 17 cancer as well. And it is possible to use all this genomic distribution of mutations to predict whether a sample is XPD mutant or not. And with that, I want to express gratitude to all the groups who make their data available, including Genomics England, and to my supervisor, Jason Wong, and thank my co-authors. Okay, thank you.
So thank you, everybody. And we've now got a few minutes for questions. So questions, if you could put them in the Q&A section down the bottom. Yes, round of applause definitely called for there. And so I'm going to kick off with some really basic and, as a non-cancer researcher, uninformed questions. So if that's okay. So what does CBS actually do? How does, because presumably you're thinking that that's actually what's driving cancers rather than XBD mutations.
Well, to be honest, we're not actually sure if the CBS mutations are necessarily oncogenic, because we've looked at recurrent ones sort of distanced to oncogenes and didn't really find anything. We think it's an artifact of the mutational processes going on in the cancer sample, and it really informs us more of the basic biology of how the genome works rather than driving cancer, I think.
Okay, all right. And does this mutational profile, apart from being associated with bladder cancer, have any clinical significance? Does it affect prognosis or treatment response or anything like that?
Well, XPD mutant cancer is associated with better response to cisplatin and a better survival. So that's partly why we want to see if we can use our model to predict whether a sample is XPD mutant or not.
Right. And so did you identify any, obviously it was fascinating looking at the mutational mechanisms and the 5FU data. Did you see anything with regard to mechanism of action of cisplatin that comes out of your analysis?
So we haven't looked at that, no.
Okay.
Yeah.
All right, so there are a couple of questions in the chat. So the first question is a compliment, great talk. What is your hypothesis about why signature 17 is not usually observed in XBD mutant and UDG mutant samples?
Yeah, that's an interesting question. Like, we know if we look at XBD mutant samples specifically in CBS, they have signature 17 in that region. We think CBS, we think is probably a combination of like unusual polymerase activity in those regions, plus the kind of genome-wide uracil process.
Okay. Benjamin, if you have more questions about that, feel free to go into the chat. Second question is from Alvin Ng. Could you elaborate more on the hypothesis of signature 17? Could you think of an approach to test uracil incorporation in signature 17 positive tumors?
We've looked at uracil incorporation in signature 17 non-CBS hotspots, and it is increased there. We've also done some experiments in the lab with a different type of chemo that also causes DUTP. So we are doing some experiments as well.
Okay. All right. So where does this research go from here? You've just touched on it a little bit. Is there any other areas that you're thinking of heading?
Well, yeah, we're really interested now in profiling uracil. We're trying to work a method up in our lab to detect it by nanopore. And actually, I didn't show it, but I looked in a lot of detail of the epigenetics with uracil, and it's very interesting. Is really associated with repression. And we want to see, does uracil actually have an epigenetic function? Or, I mean, it might just be a passive mistake, or could it regulate gene expression? That's what we're interested in.
So with nanopore sequencing, have you looked at cancer sequence data from nanopore, and does it pick up uracil? And I'm thinking then you could also get strand-specific methylation change too.
No, with our nanopore, we're still at the stage of training, training the model. We've generated training data, and our model is pretty good at calling uracil, but we need to optimize it more before we can run it on some cancer samples treated with 5-FU, for example.
Okay. All right. Excellent. Well, just to advertise a little, the NGRL has some and will have a growing amount of cancer nanopore sequence data over the next 24 months. So there'll be several hundred to low thousands of cancers that will have tumor normal data by nanopore in the NGRL that you might find of interest, and others on the call might find of interest too.
Okay, thank you.
Okay, I think we're there, and we've answered the questions in the chat. If anybody else has got any questions for Jayne, then please feel free to put them in the chat. And Jayne, I can point you in the direction of that, answer them afterwards. But otherwise, thank you very much for a really fascinating presentation.
Okay, thank you.
Okay, so we're now going to swap to Máté Manczinger, who's from Hungary, and who's going to talk with us about synergy of HLA class 1 and 2, shaping the strength of anti-tumor immune responses. And if somebody could change over from Jayne, so we're showing Máté. And again, if people put questions in the chat, that would be great, and we can go to those at the end. Thank you.
Thank you very much, and thank you for the invitation. It's a great honor of talking about our results in this great seminar, and I'm also grateful to Genomics England for providing us this very valuable data. So I'm going to talk about the HLA molecules and the role of the two classes, the combined role of the two classes in anti-tumor immune response. Most of us know that the immune system has a role in protecting against harmful substances and tolerating non-harmful substances. And in the previous several decades, it became clear that cancer is also a harmful agent against which the immune system has a very important role. And it has revolutionized our thinking about the immune system and cancer. And what mediates anti-tumor immunity? In the tumor cells, mutant peptides are constantly generated due to the nucleotide mutations. And if these peptides are presented on the surface of the tumor cells by HLA class 1 molecules, then the T cells, the CD8 positive T cells, have the potential to recognize these mutated peptides, and an immune response can be induced. And I think 20 years ago, I would be able to stop here the anti-tumor immunity thus because it was not really known that HLA class 2 has an equally important role in anti-tumor immunity, because from tumor cells, these mutated peptides are got to the lymph nodes in dendritic cells. And there is an HLA class 2 mediated presentation of mutated peptides, which is also needed for an effective anti-tumor immune response. And cancer immunotherapy exploits a mechanism which is induced or which is done by these tumor cells. They are able to express molecules, for example, this PD-1 ligand, with which they can prohibit the activation of the T cells, and the immune therapy is able to reactivate the T cells by blocking these PD-1 receptors or the PD-1 ligand molecules. And it was quite a revolution. Those individuals in late-stage cancers who had a very poor survival before, they are much more likely to survive nowadays, and this is a long survival. And the two researchers who had a great role in developing these therapies got Nobel Prize in 2018. But still around half of these patients who get cancer immunotherapy, this immune checkpoint blockade immunotherapy, they do not respond. And that's why it's essential to find out what kind of factors are important in determining an effective immune response against cancer. We focused on HLA molecules. These HLA molecules, the human leukocyte antigen molecules, are the ones that bind and present these mutated peptides on the surface of the tumor cells and also in on the dendritic cells. And they are extremely variable. They have an extremely high genetic variability. So it's very unlikely that two random individuals will carry exactly the same HLA class 1 and 2 alleles. And these different HLA variants has different peptide binding specificities. This is a binding motif here of an HLA class 1 molecule in different positions of the of the peptides. The HLA molecules are specific to different amino acids. And it has been researched, the role of HLA class 1 molecule has been researched very intensively in the previous one decade. And a lot of results reported the role of HLA class 1 genotype and the diversity in anti-tumor immune response and the response to cancer immunotherapy. There were HLA class 1 supertypes reported to have an effect on response, the HLA homozygosity, the evolutionary divergence of HLA class 1 molecules. We also reported that the breadth of peptides that are bound by HLA class 1 molecules is very important in determining anti-tumor immune response. Also, the AO3 supertype of HLA class 1 molecules has an effect on anti-tumor immune response. And it was already suggested that the MHC class 1 genotype restricts or determines what kind of oncogenic mutations can remain in cancer cells. However, the upcoming publications suggested that these results are not always robust. So, for example, it was shown that the HLA class 1 diversity, the homozygosity, the evolutionary divergence, and the supertypes doesn't have a solid effect in pembrolizumab treated cohorts. Also, it was shown that the HLA genotype, that it was suggested that it is an important effect which kind of oncogenic mutations are remaining in tumors, but it was more likely an artifact, a statistical artifact. And also there was a large meta-analysis carried out by Charles Swenton and his colleagues. And in this meta-analysis, they did not find an effect for HLA class 1 related features. In parallel, evidence accumulated about the role of MHC class 2 molecules in anti-tumor immunity. It was shown that without MHC or HLA class 2 mediated activation of CD4 positive T cells, it is the anti-tumor immune response cannot develop. And in an analogous way, HLA class 2 immunogenic mutation burden predicted the immune checkpoint blockade response similarly to HLA class 1 mutational burden. And what we thought that these inconsistent results might be partially caused by examining the two classes separately. So we thought that in several cohorts, the effect of a given HLA class 1 or an HLA superclass can be seen because the HLA class 2 background is ideal. In other cohorts, the HLA class 2 background is not ideal. So we thought that there might be some HLA class 1 and class 2 allele combinations that are based on their binding preferences, more likely to bind neopeptides generated in a given tumor. And when they occur together, their effect will be much different when they occur solely or separately. And we will see the real effect when we consider both variants, both the HLA class 1 and class 2 variants. And we examined the melanoma malignum tumors in our study. And we collected ICB-treated melanoma patients, and we carried out survival analysis. We built Cox models, and in these Cox models, we integrated an interaction term between different HLA class 1 and class 2 variants. So the model consisted the presence of a given HLA class 1 variant, a given HLA class 2 variant, and the interaction effect between these variants. This interaction term shows us how is for example, the HLA class 1 molecule effect is modified when the HLA class 2 is present in the tumor. And what we have seen, we have identified 9 combinations when the effect of HLA class 2 and HLA class 1 together was much different than separately. On this plot on the right side, there is a positive hazard ratio, meaning worse survival, and on the left side, there is a negative hazard ratio, meaning better survival. And as you can see in several combinations, in 2 combinations, the joint effect of the HLA class 1 and class 2 is beneficial, while their separate effect is much more different. And we found 7 interactions which were more detrimental. So when these alleles occurred together, the class 1 and class 2 allele occurred together, they had a very detrimental effect on the survival of the patient. So on an example, the A0101 and DRB10101 combination, as you can see, when only the A0101 is present, or the DRB0101 is present, they don't really have an effect, but when they occur together, they have a very detrimental effect on patient survival. And next, we classified patients based on whether they carry these detrimental combinations or beneficial combinations or none of them. And we found that those individuals that carry beneficial combinations, they are much more likely to survive, and those individuals that carry detrimental combinations, they are unlikely to survive, and it's independent of the treatment type. Next, we wanted to see whether this phenomenon can be seen in ICB-treated patients only, or also in treatment-naive patients. So we examined the survival of treatment-naive patients, and we saw no effect on the survival of melanoma patients if they were in stage 1 or stage 2. But when we examined patients who already had metastasis, so the tumor was progressed into at least a lymph node metastatic phase, then the same trend was visible. The beneficial the patients carrying beneficial combinations, they had a much better survival than the others. And it was also seen in the Genomics England melanoma patients. And we wanted to know, okay, we can see that there are detrimental combinations and beneficial combinations, and there is a difference between the survival of patients if they are diagnosed in an early stage or in a late stage. it suggested to us that something is happening during progression in those individuals who carry detrimental and beneficial combinations. So we aimed to analyze the immune phenotype of primary and metastatic samples separately. So I will show you the results for primary samples first. Here on the left side of the plots, you can see the CD8 positive and B cell count in the tumors carrying detrimental, neutral, and beneficial combinations. And as you can see, in primary tumors, the CD8 positive T cells and B cells are already elevated if these samples carry detrimental combinations. However, if these samples carry beneficial combinations, both the CD8 positive T cell count and the B cell count is lower than in the other groups. In metastatic state, we did not see these differences between the groups. We also checked the histologic report for tumors in the Genomics England cohort, the primary tumors of the Genomics England cohort. And there is a phenomenon in melanoma called regression. Regression means that in the primary tumor, there is a very strong immune response against the tumor, which will result in the absence or the destruction of a small part of the tumor, and there will be no tumor in that region. So it can be defined with the pathological examination. And what we have seen that in those samples which carried the detrimental combinations, the regression was more common, especially if the tumor was thinner than 2 millimeters. So it also suggested that there is a highly active immune response against the tumor in the detrimental group. We also showed, I don't want to show too much figures, so we also showed elevated NK cells, elevated cytotoxicity score, and a generally inflamed phenotype in the primary tumors with detrimental combinations. However, in metastasis, we expected to see something different. In terms of immune phenotype, we did not see too much difference, but we expected that there might be some immune evasion, irreversible immune evasion that has been developed in these samples, these metastatic samples carrying detrimental combinations. And we found in case of the detrimental combinations that many tumors were associated with copy number alterations of neoantigens. Also, these tumors downregulated on the RNA level these neoantigens, and this might suggest some immune evasion in these tumors. And we also aimed to test how strong is the immunological selection in these tumors. And we have seen that the immunological selection was released in these tumors, these metastatic samples carrying detrimental combinations. On this plot, you can see that a lot of neoantigens were effectively presented by HLA molecules in these samples. So it suggests that a lot of immunogenic mutations were able to accumulate in these samples carrying detrimental combinations. And also this in evolutionary biology, this DNDS is used to assess the selection on the tumor. And we also determined this expected and observed number of neoantigens, and what we have seen that it's also elevated in case of detrimental combinations. So we have seen that detrimental combinations has this biphasic effect during tumor progression. It's associated with an active immune response in primary samples, and immune evasion is induced in metastatic samples, which might explain the worse patient survival. While beneficial combinations, they are not associated with an active immune response in the primary samples, but potentially more active immune response in the metastatic samples, resulting in better patient survival. And we wanted to explain what kind of peptide binding properties of these combinations might explain these findings. So in the beginning, in the hypothesis slide, I suggested that when an HLA class 1 and HLA class 2 molecule has a similar specificity, it might explain why the immune response against the tumor can be more effective, because both classes, from both classes, there is an activation of the immune response. And we examined the peptide binding specificities of HLA class 1 and class 2 molecules in the detrimental combinations and the beneficial combinations compared to the neutral ones. And here there is an example with a highly similar peptide binding specificities. On the left side, there is the HLA class 1 molecule. On the right side, there is the HLA class 2 molecule, and they have similar specificities in different positions, as you can see. As an opposite case, there is a class 1 and class 2 molecule with absolutely different peptide binding specificities. And what we have found, you can see the distance between the different specificities of HLA class 1 and class 2 molecules in detrimental, neutral, and beneficial combinations. And as you can see, both the detrimental and the beneficial combinations has smaller distance between the peptide binding specificities than the neutral ones. But what makes them different? So both the detrimental and the beneficial HLA class 1 and 2 molecules in these combinations has similar specificities, but then what is the difference? And we here on this plot, you can see the composition of melanoma tumors, what kind of mutations, substitutions, amino acid substitutions, what is the distribution of these substitutions in melanoma tumors. And as you can see, there are several amino acid substitution types that are quite common in these tumors. And we found that the beneficial combinations were more specific for the serine phenylalanine substitution, while the detrimental combinations were more specific for the glutamic acid and lysine substitution. And this glutamic acid lysine substitution was suggested to be a radical mutation, a highly immunogenic mutation, which makes or which changes the structure of the peptide highly, and that's why it will be more immunogenic. So it seems that the detrimental combinations are more likely to present highly immunogenic mutations in the early phase, while the beneficial ones are present less immunogenic mutations, so they are probably less likely to induce immune escape. So as a summary, in case of detrimental HLA combinations, we have seen that as they present these immunogenic mutations in the primary tumor, there will be an intensive inflammation that will lead to immune evasion in metastasis. However, in the case of beneficial HLA combinations, in primary tumors, there is not a intensively cytotoxic immune response. There are other kind of immune cells in the primary tumors of beneficial combinations, but in metastasis, there will be an effective and intensive cytotoxic immune response. As a conclusion, I can say that we found that HLA class 1 and 2 together has a much stronger predictive and prognostic effect than when we examine them separately. I only can repeat myself that detrimental combinations present radical mutations leading to immune evasion, beneficial combinations present less immunogenic mutations, and an effective immune response is found in late-stage tumors. Our results can be further read in a preprint on bioRxiv. And I would like to thank everybody who helped in this project. Thank you.
Okay, thank you, Máté. That was a very dense and detailed, excellent presentation. For questions, again, please put the questions in the chat box down the bottom. There are some questions that are coming in, but I'm going to take Chairman's prerogative and just kick off by just asking. So one question would be, is there any personalized cancer vaccine data to back up what you're seeing? Because that would, I would imagine, give you pretty good direct evidence that it was the specific HLA type, because the vaccines are designed around specific HLA allele presentation capabilities?
Yes, it would be a great analysis to test this on neoantigen vaccine data. I think that yet there is no public data, or there are no public data available, but hopefully it will arrive soon. So yes, it's a great idea.
All right, so the next question I had was about how you actually analysed the HLA class 1 and class 2 alleles, and whether they were analysed with taking into account whether they are in cis on the same strand or in trans on separate strands. The reason being that if they're in cis, then basically it's not really clear from what I can see how you would distinguish between just a third HLA gene being responsible for the observation versus it actually being a combination.
Yeah, it's a great question. So it was already a quite complicated analysis. So if we would, we wanted to enlarge this examination to examine not only two combinations of one HLA class 1 and class 2 molecule, but other taking into account longer haplotypes. But based on the short or the small sample size, it was not really available. And this cis-trans, I don't know what you mean. So you mean that on the same strand?
Are they on the same strand or not? Now, you might not have that haplotype data, of course, but
yeah. So it should be quite important in case of HLA class 2 molecules. But as I know, it has been suggested that trans and cis HLA DP and DQ combinations are also occurring in the tumors. So what we have done is simply generated all kind of combinations in the tumor samples. So we didn't differentiate between them.
So it's not impossible that this is actually due to a third site that's actually just in linkage disequilibrium with one of the other two.
Yeah, it would be, I think, quite hard to control for all these effects, but you are right. Yeah.
And so related to that, you presented data about CD8 T cells and its interaction with this. But of course, class 2 tends to present to CD4 T cells. So how do you see any difference in CD4 infiltrates or activation in your ones which have got the combination of class 1 and class 2?
Yes, I missed it from the slide, but the CD4 was also elevated in these samples.
Okay. All right, thank you. So then there's a question in the chat from Anonymous who asks about correction of sample size on the interactions between class 1 and class 2. Obviously a massive number of interactions, and it was good to see that you did a replication, but did you try to correct it all?
Yeah, it's a great question. So based on the limited sample size in the cohort, what we have done, we checked the result on a validation cohort too. So this is what we were able to do, but this limited size of the cohort made it not really possible. Yeah.
Okay, so to some extent, this is a discovery program. I imagine that, you know, this is always a problem with HLA association studies anyway, so you're not alone in facing this challenge, but at least you did see some replication, which was nice. So I don't see any other questions in the chat, in which case I'll just ask at the end, where does this lead from here? What are your plans from here with the research?
And so what we aim to do, these ICB cohorts were HLA monotherapy, or monotherapy cohorts. So they got either CTLA4 or PD-1 inhibitory drugs. And we expect to find different effects when the patients get combined drugs, as the HLA class 1 is suggested to be important in CD8 positive and in PD-1 blockade. So the PD-1 blockade suggested to act on the level of CD8 positive T cells, and HLA class 1 molecules have an important role in presenting peptides to the CD8 positive T cells, and the CTLA4 blockade suggested to have an effect on CD4 positive T cells and HLA class 2 presence to these cells. And what we expect that there might be different results. So these specific results might be stronger or weaker. We don't know, but this is in our plan
according to the treatment.
Check. Yeah.
Okay. All right. Máté, thank you so much. That was an outstanding presentation. So we'll close up there, and I'd just like to wish everybody a happy Christmas. Yes, that's right. It's just under a month away, and Happy New Year. And we look forward to seeing everybody again on the 30th of January, last Tuesday in the month, 2 o'clock. Bye then.